The recombinant DNA is the alter of genes in a particular genome. The organism containing recombinant DNA is called recombinant. Which organism carrying the recombinant DNA molecules, that is called recombinant. In recombinant DNA technology, a desired gene, for example, human insulin gene, is transferred to a plasmid. Plasmid is to act as a vector. It is a double-stranded extra chromosomal DNA present in the prokaryotes. The plasmid containing insulin gene is the recombinant DNA. The recombinant DNA introduced into the bacterium such as E. coli. The E. coli is called recombinant organism. When E. coli multiplies, the human insulin gene also replicate and produce multiple copies of insulin gene. So the result is the more production of human insulin gene inside the E. coli bacterium. This is also called gene cloning. Then steps for recombinant DNA technology. First, preparation of desired gene. The preparation of desired gene from natural DNA or mRNA. mRNA is nothing but messenger RNA from the source. Then second step is isolation of DNA. Isolation of plasmid. Plasmid is a vector to act as a vector. A plasmid is a circular double-stranded DNA found in the bacterial cell. Then the third step is insertion of desired gene. Our desired gene is human insulin gene. The desired gene is inserted into a plasmid. That is a vector. Yep. The plasmid is cut with a restriction enzyme. That is our desired gene, the human insulin gene, cut by the restriction enzyme to the restriction enzyme, and finally the formation of recombinant DNA. Restriction enzyme it is also act as a is called molecular scissor or molecular knife. Then the steps for recombinant DNA technology clearly show the figure shows the clear diagrammatic structure for recombinant DNA technology. First, the phase human, we take human mammalian gland produced the pancreatic cell produce human insulin gene. The human insulin gene producing the gene insulin gene is our desired DNA. The desired DNA cut by restriction enzyme. Restriction enzyme otherwise called molecular scissor or molecular knife or chemical cutter. The fragmented DNA of desired gene is placed or inserted into the plasmid. Plasmid is synthesized from bacterium. Then our fragmented desired DNA to inserted into the plasmid by paste or seal by the enzyme of DNA ligase. The DNA ligase enzyme to act as a sealing the fragmented DNA. So the final product recombinant DNA produced. The final product of recombinant DNA is inserted into the host organism, like that prokaryotic organism in bacteria, Escherichia coli, shortly E. coli bacteria. The E. coli bacteria is a host organism. The host organism the replicate the human insulin gene multiple copies. So our desired gene, the insulin gene, produced or multiply in large copies. So the recombinant DNA play a vital role in genetic engineering. Then application of recombinant DNA technology. First of all, the application, the production of insulin, human growth hormone, interferon, enzymes, steroid hormones. So large molecules are produced by recombinant DNA technology. Then industrial use, the alcoholic beverages, organic acids, amino acids, and biogas produced from the bio plants. Then next application is the rDNA produce recombinant DNA play control the pollution by the chemicals applying by the insecticide or pesticide. um, um, so the rDNA technology to reduce the pollution caused by the chemicals. Then the diagnostic purpose, recombinant DNA technology to clear the diagnostic the particular disease or pathogen. For example, sickle cell anemia in the jar recessive gene are identified, and hemophilia, diabetes, Lisa Nachman syndrome. So the syndrome, the hereditary disease like that, hemophilia, sickle cell anemia, are identified uh, by that recombinant DNA technology. So the recombinant DNA technology, it's also help the chromosomal mapping, like that human genome project. And finally, the rDNA technology is also used for production of vaccine, like that hepatitis, liver cancer, rabies, cholera, and malaria, etc. So the recombinant DNA play a vital role in genetic engineering technology. It is a very important topic. Thank you for watching this video. Thank you.